Hi, this is Dr. Ben Morrill. Welcome to episode 3 of Reptile Genetics Weekly. We've been updating again, lighting and sound and video stuff. So hopefully I look a little less like an Oompa Loompa this time as I did last time. I did actually spend a lot of yesterday out in the sun, so I do have some natural color, but hopefully I'm not as orange as last time. So wanted to start off this time again with an update for people that have samples here. So today is April 24th. And so right now, Sean is putting together all of the samples we've had come in over the last couple of weeks. So if you have your sheds received as of the 24th of April, then your sheds are going to go on this run that we will have results likely mid to late next week. So I guess that would be late April. The last little bit of April or 1st of May, we'll have the next batch of results. So if your sheds, if you don't have results yet, but you do have sheds received by the 24th of April, and that's when you'll likely get your results, whether that's for sex determination or for ball python morph tests. All right. So what have we got for questions, Kayla?
I don't know. Let's see. Pulling them up here now. All right, so first question we've got today is it possible that the desert ghost shed issues that we've had could be from some DG samples being enhancer genes without it being known, or other unknown lines of DG? Or since they are compatible or one and the same, would they show up the same on those tests? And this is from Tim Slotkey on Facebook.
All right, thanks for the question, Tim. So this is a good question, and it's something that people have talked about some. Here's a YouTube video that covers kind of this question: if enhancer or desert ghost are different. Got some good pictures there, what they look like, and then here's a couple more examples that are on YouTube of people discussing these topics. So yeah, it's something that people have recognized. They obviously look very similar, but they came from different breeders, different origins, and wondered if it's the same thing. So what I can tell you from the desert ghost testing that I've done so far, when I have run enhancers, they do match identical to desert ghost. That being said, I don't have for sure the mutation that causes desert ghost. So this is subject to change, but these are mutations that travel the majority of the time with desert ghost. So my guess is that from what we can see so far, the actual mutation that causes desert ghost will probably be identical to what causes a enhancer. So they are likely the exact same thing, just two different places where it was figured out that you could breed the animals that were hets and get these really awesome looking enhancers or desert ghosts. But like I said, it could be subject to change. We hopefully will know for sure sometime in the next month or so. We'll be able to have a test that gives us over 99% accuracy for desert ghost and enhancer, and then we'll be able to have that be available for people as a service for pos het desert ghost and pos het enhancers. So at that point, we'll know for sure if they are indeed identical or if there is actually two different things going on. But as far as our test right now being 85% accurate, the question of, so does that mean that maybe enhancer makes up that other 15%? What I can see right now when we do desert ghost tests, what it looks like to me is that we have a mutation that's very close in proximity to the actual mutation that causes desert ghost, but it's not the actual one. So that would be different than there being two different mutations that cause. So like with yellow belly, there's two different mutations that cause yellow belly that are close to each other. And so you have to actually find both of them and test for both of them to be able to tell if an animal is yellow belly. Well, with desert ghost, what I think is happening is we have a mutation close to it that doesn't cause it that we're testing right now. And most of the time it travels the same, just like with banana and sex, the sex determining, whatever the sex determining gene or genes are. Most of the time, banana or coral glow travels together with that sex determining gene or genes. 90% of the time, 10% of the time, crossing over happens in between those two. And then you can have that, 1 out of 10 that if they're supposed to be male bananas, if it was a male maker male, so most of your bananas are going to be males, but the one female that pops up, that's because there was a crossover event in between. And so that's what I think is going on with this 85% with Desert Ghost is we have a mutation, a marker for Desert Ghost that's just not quite the precise one that's causing it, but it's close in proximity. And so most of the time it travels together and would still give us the same answer, but sometimes it does get split up from that actual mutation that causes Desert Ghost. So it's a good question. I hope that helps make it a little more clear.
That's cool. Thank you for answering. Let's see. We've got next a question from Necromancer Morphs on Instagram. Questions. For ball pythons, would you be able to do ROH, regions of homozygosity, testing to see how closely related the parents were?
Good question. So yeah, we're very interested in being able to tell how closely related animals are to each other. So the way we are going about that is a genetic identity test where we're looking at actual sequence and we're looking at the whole genome. So we don't need a really, really deep coverage that's really expensive. We're trying to optimize a way to have low coverage, but still be able to have enough genetic information about an individual to tell it apart from all other individuals. So it'd be a unique set of, right now, where we have it with ball pythons, we have about 10,000 markers that allow us to be able to figure out how animals are related to each other. And so what this is exactly like, which, we had the screen pop up for a second there. Go ahead and go to that ancestry.com, Kayla.
Got ahead myself there.
She foreshadowed for us. So ancestry.com uses the same thing. So when you go on there and you submit a DNA test, they're able to use these thousands and thousands of markers in humans that are used to be able to tell relatedness and groups that are, part of a very tight group like a family, or a looser group like from a geographic region or something like that, or descendants from a specific king or something like that. So the same technology was used to help I think I talked about this in a previous episode a little bit, but the Golden State Killer was caught this way as well. They're able to use information like on ancestry.com. They took the DNA from the crime scene and they uploaded that to ancestry.com, websites like it, and they were able to find someone that they knew was a relative, a familial match to the crime scene DNA. And they had to go back a few generations, I think 4 generations back, and then work forward. And that they knew that 4 generations back was that common ancestor. And then they worked forward and looked for males that were about the right age. And they're able to narrow it down to the person that ended up getting arrested for that. So it's a pretty powerful technology that can be used to tell relatedness, differences, group people or animals in various sizes of groups, like I said, to tell 2 very closely related individuals apart, or to tell people that had a ancestry that started out in England versus France or something like that. There's a lot of different comparisons. Once you have a lot of genetic information in your database, you can make a lot of different comparisons and say some cool things. So the things like the dog DNA test, the cat DNA test, all those kinds of things, they're based on similar markers that can be found that a high percentage of the time are part of this group or that group, and that can help tell a story. So specifically for us, we would like to be able to use it for parentage testing, would be one reason. And then another one would be, I guess with parentage testing, if you have 2 males or 3 males in with a female, and you could do a paternity test and figure out for each baby who the daddy was. And then also in dry marcon, or the indigo snakes, the keepers and breeders for them are very interested in ensuring they have as much genetic diversity as possible to try to avoid any inbreeding problems, inbreeding depression. And then also this same technology can be used with panther chameleons to be able to test for locality. So those are all things that we're working on right now to develop tests and use a similar kind of idea that you're bringing up with the regions of heterozygot or homozygosity. Same thing. We're doing it at the sequence level, the A's, T's, C's, and G's, finding those genetic differences and being able to make comparisons between groups and within groups, to be able to do those kinds of things, parentage testing, all that we were just talking about. We'll have that capability first of all in ball pythons, and then some of these others we'll continue to work on. And this last week, actually a couple days ago, I got to go to the AZA herpetological tag meeting and talk to zookeepers about how this could help with their stud books to help ensure that when they're deciding on breeding pairs, that they also are able to have as much genetic variation as possible, and to see if there are reasons to group animals into smaller subspecies, or to split a species into 2 subspecies or something like that. There's different things that we can do once we know that much information about an individual or a group of individuals. And that's all possible from the DNA, from the historic information that's stored in the DNA. Pretty fun stuff.
That's awesome. So our next question is, how can we contribute to sheds of snakes with known morphs to help with developing tests? For example, known hets or visual recessives. And that's also from Necromancer Morphs on Instagram.
All right. So this is a question that I love because we are always looking for help here. Definitely everything that we do starts out with us being able to do a whole bunch of sequencing on sheds from animals where we know what their genetics are. So to make a test to be able to tell a difference whether something is het for a trait or not, let's say het sunset and ball pythons, we have to have a big group of sunsets and het sunsets that we know for sure what they are, and then a big group that we know do not have sunset or het sunset in them at all. And we do a whole bunch of sequencing, billions and billions of base pairs from each, and we use software and powerful computers to be able to compare those. And that way we can go through those comparisons and figure out what that one difference is, whether it's a deletion or base change or whatever it is that causes sunset. And then we can develop a test. And so it's a big, very complicated process that we use a lot of cool technology that many people have developed over, especially the last 10 years. We've made a lot of progress in being able to make comparisons like this with sequence from 2 different pools and tell a difference, and then be able to design a test. But really, all of that we're doing, the complicated things, it has to start with us having a whole bunch of sheds. So if you're to go to our website on like a computer or a laptop, this is what you would see. And if you go where the arrow is there at the top, it says help us. And so when you go there, it gives you some information on how to send sheds. So if you have a ball python morph like puzzle, if you have puzzle ball pythons and you'd like there to be a puzzle test, or you have albino boas and hypoboas, you'd like there to be some boa mutation tests, or maybe you have anacondas and you want baby anacondas to be able to have a sex determination test. But that was something we talked about at the herpetology tag meeting that could potentially be helpful, because some species are more difficult to tell as babies what gender they are until they've grown up some. So that's another place where we can use sheds coming in if it's a new species for sex determination. So any of those things, we can use sheds coming in. And so this is how you can do that. If you're on a cell phone and go to our website, we have just a quick screen recording here. Go to the Help Us and the drop down. It takes you to the same page. As you scroll down, you can get that same PO box to send sheds to. So right now, those come directly to me. And so I take those sheds, I send them, we pay money and spend time analyzing pretty much everything that comes through. It might not be immediately. I might need to get enough, like if it's for a sex determination for a very small group of animal, maybe it's a species that not very many people have. It might be a little while before I have enough to work with that, but we definitely catalog those and use those at some point to be able to design tests. So please do reach out, let us know, send sheds in. We definitely use them all the time. And really, keep in mind, it all starts with you all. We have to have all these sheds come in. So here's a good representation of me buried in sheds coming in. And you can tell I'm happy. This is what I like to see. I was telling Kayla, just in this little room that I'm in right now where I'm recording, I have 3 boxes. They're probably, I don't know, 2.5 by 2.5 by 2.5, pretty good size feet, pretty good size boxes. I have 3 of those completely packed full. This is just a few that I pulled out. But we've got lots and lots of sheds from lots of people all over, and very thankful for that. And that's what's made it possible to get to where we are now. And people that are paying for the services that we have available now, the sex determination tests we do have for ball pythons and colubrids, and the morph tests we have available for ball pythons, a very big part of that money goes right back into designing more reptile genetic tests. So it's a very high percentage of your cost when you pay for a test is going to go right back into designing more. So that more and more people can send in more and more sheds, and I can be there happy with piles of sheds and sending them off for sequencing. But it does definitely all start with you all. So please do, if you've got shed. I mentioned at the herpetological tag meeting yesterday for these zookeepers, I told them, even if you don't plan, if you have some reptiles and you don't plan on doing any kind of testing that's available right now, the fact that you can take a shed from them today, as long as you remove it quickly after it's shed, dry it out completely, put it in a Ziploc bag or an envelope, store it in a dry place at room temperature is completely fine. That shed will stay good for years. I've run them after over 5 years and they've worked perfectly fine. So even if you're not necessarily going to use it right now, even if you're not going to send it to me to start playing with right now, if you at least take sheds from each of your animals that you have in your collection, at some point in the future, that shed can be used, whether it's with me or with a test that eventually gets developed. You can have that animal tested, or it could be part of being able to build a pedigree for animals that you have, or be able to tell whether an animal you had 5 years ago is actually related to one you end up getting later, or one that someone else has. So there's a lot of things that you can do if you just keep those sheds. If you, instead of just taking it out of the cage and throwing it away, if you take it out of the cage, rip a little piece off, throw the rest of the way, and then save that little piece and just write on there the animal name or number so that you can, refer back later and know what animal it's from. And that shed has a lot of really interesting historical information that we can access. And each shed that comes in and we do the sequencing and we have that information banked, it gives us more and more opportunities to be able to tell cool stories about these animals that we all keep and are obsessed with. So it's part of the fun, and we're building that more and more each month that goes by. We're getting more and more sheds coming in, and thank you to all of you that are sending them in for testing that you're paying for, or just sending them in to develop new ones. That's what keeps us going at Rare Genetics Inc.
Mm-hmm. All right. So do we have any final thoughts?
I think that's it. Thanks for the questions. Thanks again, those of you that have liked, subscribed, put comments in the YouTube comment section or on Facebook or Instagram. We appreciate that. And once again, like I said before, that's where the content will come each week for us as we do a Reptile Genetics Weekly episode. Kayla and I go through those and talk about them almost every day, but definitely 2-3 times a week at least as we prepare for the show. And so you can make a difference if there's something you're interested in or wondering about. Send questions in and make comments, and we look forward to making more episodes.
Absolutely. Thank you, guys. I guess that's a wrap for us today.
I think so. Thanks, Kayla.
All right. Sounds good. See y'all next week. And outro.